Hello dear viewers, myself Nasir and you're watching Mr.Zoologist YouTube channel. Today we are going to discuss about toxicity testing. Toxicity testing is a systemic evaluation of the toxic effects that an agent can produce. In actual, they are not capable of demonstrating absolute safety, but that can determine how much harm can be done or is likely to be done. Toxicity tests are experiments or trials designed to assess or evaluate the dose or concentration of a toxicant and the duration of exposure required to produce negative effect or harmful effects. Toxicity tests compare the response of an organism exposed to a specific chemical at various concentrations to the response of the same organism's chemical. Toxicity tests are mostly used to examine the specific adverse effects or specific endpoints such as cancer, cardiotoxicity, and skin or eye irritation. Toxicity testing also helps to calculate the no observed adverse effect level, that is also known as NOIL, and it's helpful for clinical trials. Toxicity tests characterize the toxicity and the level of toxicity. They help to determine the dose response and the target. Toxicity tests are performed to assess the safety or hazards of several substances such as industrial, chemicals, pharmaceuticals, and consumer care products that can vary according to the market or demand. Toxicological screening is very important for development of new drugs and for the extension of therapeutic potential of existing molecules. If we talk about different toxicity tests differ from each other, first of all is acute toxicity testing, the second one is subacute toxicity testing, the third one is subchronic toxicity testing, and the fourth one is chronic toxicity testing. Certain toxicity tests such as teratogenicity, mutagenicity, reproductive toxicity, carcinogenicity, immunotoxicity, and neurotoxicity evaluate certain type of toxicity in the individuals. Many of current toxicity tests use laboratory animals such as rats, rabbits, and rodents. Animals are useful models to predict the toxicity of chemicals in humans because they have similar cell organelles, cells, and organs. If model is not close to human, uncertainty of results will increase. And if you talk about acute toxicity testing, study the adverse effect of single dose on particular animal species, that will be single exposure. If we talk about its duration, or duration for the acute toxicity testing includes 14 days, usually carried out with two different animal species, one is rodent and the other is non-rodent. Results are represented in terms of LD50. Investigational product is administered at different dose level and effect is observed for 14 days. Duration or experiment during the experimental period, all mortalities, morphological and biochemical and histological changes are observed. In subacute toxicity testing, it is also known as short-term toxicity. It involves rapid administration, usually on daily or 5 times per week basis. Duration period is about 28 days. One or more species are used. It is used to assess the nature of effects. And if we talk about subchronic toxicity testing, rodent and non-rodent species are used to determine the subchronic toxicity. Duration is of 3 months. Test substance is administered orally for 90 days, repeatedly, and regular body weight variation, biochemical and cardiovascular parameter changes, behavioral and histological changes are observed carefully. It is used to determine the maximum tolerable dose of toxicity. And if we talk about chronic toxicity testing, also known as long-term but definite toxicity, it involves rapid administration over a considerable period of lifespan of test animal. Its duration is about 3 months to 2 years. Multiple species can be used to carry out toxicity testing, chronic toxicity testing. It measures dose, high dose, medium dose, and low dose levels as well. During study period, animals are observed for normal physiological functions, behavioral variations, and alterations in biochemical parameters. All these are observed at regular intervals, at least after every 14 days, not less than 14 days. At the end of study, tissues are collected from all over the body and subjected to histological analysis. It is used to study the secondary effects such as carcinogenicity. It also evaluates safe levels of toxicants. What are experimental design for toxicity test? First of all is a selection of animal species. Second one is route of administration. Third one is dosage, and fourth one is observation and examination. If we talk about selection of animal species, Choice of test species for toxicity testing is often affected by economic considerations. Some people do not afford experimental animals. Limitation of laboratory facilities or problems associated with real target species. Two rodent species are most often used for study: the laboratory rat and the laboratory mice. Many species of birds are vulnerable to intoxication, including starlings and pigeons. And if we talk about route of administration, the toxicant should be administered through the route by which they are exposed. Third, the dosage refers to administration of toxicant in doses. Different types of doses are given, like as in milligram, in ppm, parts per million, parts per billion, and parts per trillion. And if we talk about observation and examinations after administering the toxicant, they should not be only examined for number and time of death, but the examination should be done for autonomic and behavioral effects. Autopsy and post-mortem examination are done to detect histological abnormalities. That was the video for today. Hope so you have enjoyed it. We will meet you in the next video. Till then, peace out.